our discussion, so you must have already learned one thing, that initially there were two germ layer present. It was called as a bilaminar germ disk. Now, if you remember that, I'm just telling in short. Suppose you have got or the trophoblast cells. You remember there were the trophoblast cells present, and there was the inner cell mass present. You remember there was inner cell mass also present. The
Yes,
inner
sir.
cell mass were connected by gap junction, and the trophoblast cells were connected by tight junction. Remember that?
Sir, ওটা ভুলে গেছি স্যার।
Acha, okay. So always remember that if this process is called as compaction. Okay, now suppose I talk about compaction process, and when is compaction process happening? This is happening when the entire conceptus is actually moving towards your uterus. Okay, not your uterus but any like pregnant uterus. It is moving towards the uterus from the oviduct. Okay, so from the oviduct, when it is moving towards the uterus, and if you understand, that is the moment when progesterone level is very very high inside the mother's body, because it is the it is happening post fertilization, mane post 14 days. So after 14
Yes,
days
sir.
of a menstrual cycle, which hormone is high in the female body? It is progesterone.
Cholesterol.
Yes. So if progesterone is there, so naturally progesterone, because what will it do? It will cause endometrium to swell up. It will cause endometrium to secrete. The endometrial glands are going to secrete a large amount of fluid, and the uterus will be loaded with fluid. Okay, there will be fluid everywhere. So what will happen? The cells which were present initially, it was just a cluster of cells. Okay, there were like 16 to 32 cell stage, which was called as morula. Okay, so then there was early morula, late morula, and all those things, and then it dropped in that water, dropped in that liquid, dropped in the fluid of the uterus. Now what happened? Gradually fluid started to move in between the cells. The fluid started to percolate or seep within the cells, and it started separating the cell, because there was a ball of cells present. Now inside that ball, when slowly and slowly fluid will enter in, so it will try to create separation between the cells. When it was
Yes, sir.
separating, it was found out that some cells which are cordoning the outer side, some cells which are surrounding the outermost side, those cells are not opening up. It's not like they're bursting. The ball is not bursting. Why the ball is not bursting? The ball is not bursting because in between these cells which are forming the outer periphery, in between these cells there is present, what is present here? There is present tight junction. Okay, these are the parts where tight junction is present between every cell. I'm not showing in all, but between each and every cell there will be tight junctions present. So because of these tight junctions, the cells are not detaching apart. Now the inner cells, some cells are present who form again a cluster here. Now these cells are going to form a cluster. Suppose I draw 3 cells here. Now what you have to understand is, though here I have drawn 3 cells, but there will be not, of course there will be not 3 cells, there will be many cells, and they have gap junctions between them. Okay, they have to communicate with each other, because later on this inner cell mass will give rise to the embryo, and hence we will call it as embryoblast also. Trophoblast, nutrition. Okay, maintenance. So trophoblast will help in maintenance bolo to protection. Okay, so it will protect that embryo and it will form a covering outside. So this outer cell mass is called as trophoblast, whereas the inner cell mass will be called as the embryoblast. Okay, so this is the basic idea. Now if I have to tell in just short, because our discussion is not this really. So okay, so next what I can tell you is, if you move over here, so next step that you are going to see, so next step that you are going to see, that is these cells, okay, there is a space present over here, there is this space present over here. So this space is called as blastocele. Now why it is called as blastocele? Because you see here the division that takes place, that is mitosis like, but it is not really mitosis, so it is a cleavage division. And the way we know that after mitosis there are one cell gives rise to two daughter cells. Similarly, here you will see that one such cell, when it divides, we don't call it as daughter cells, we call it as blastomeres. So when things are happening in a blastomere level, so we are going to call it as blastula or blastocyst. What is cyst? Cyst is any swelling in your body which is filled with fluid. Any fluid filled swelling is a cyst. So if here there is fluid accumulation, it's a swelling having fluid inside it, so we can call it as a blastocyst. And this space, this is called as blastocele. Okay, so these are some basic data. Now suppose I show you in this way, that suppose this is one, these are the trophoblast, okay, this is the lining of trophoblast, and inside there are inner cell mass present. So these are the inner cell mass present. Okay, these are the inner cell mass present. Now what I want you to understand is that the inner cell mass which is facing the blastocele. Okay, so let me put it in this way, then I think it will be clear to you that you see these are some inner cell mass cells which are facing the blastocele, the green one. See, here the green and the red both are ICM, both are inner cell mass, right? But why am I demarcating them with green? Because I want you to focus on those inner cell mass cells which are actually present adjacent to the blastocele. Are you getting my point?
Yes, sir.
So why I want you to focus there? Because you will understand now that these inner cell mass which are facing the blastocele, they become cuboidal in shape. Okay, they are becoming cuboidal in shape. I'm not making it accurate, but yes, they will be definitely cuboidal cells, central nucleus, whatever you have studied in histology, the way cuboidal cells are. And the rest of the cells are going to become columnar. Now, when these rest of the cells are trying to become columnar over here, in between them, see there are details over here, but since this is not our topic today, our topic is basically notogenesis, so I'm just keeping it brief. So here in between these columnar cells, see I'm drawing all the cells as columnar. Nucleus is towards the basal pole. So there was fluid accumulation going on. Okay, like this. See, I have changed the right hand picture as well. Can you see the fluid accumulation going on in between?
Yes, sir.
Yes.
Yes, sir.
Now this fluid filled space is called as the amniotic sac. So basically these cells are called as amnioblast, and these cells which are like cuboidal, they are present
Hypoblast.
below. Yes, they are hypoblast, and if below is hypo, then above is
Epiblast.
Yes, correct. So this is the epiblast, and this is the amniotic cavity.
Cavity.
Next step, what happens? The next step is that suppose this is the thing, this is the trophoblast, and these layer, like suppose this is the hypoblast. Now hypoblast gives rise to another layer all along this area. Okay, epiblast remains over here, that is not a problem. Epiblast definitely stays here. Amniotic cavity stays over here, but do you realize that the blastocele is gone? Because now blastocele is lined by a layer. We cannot call it blastocele anymore. because by definition
Yes, sir.
blastocele was lined by trophoblast on one side, but now it is no more. So we give it a new name. It is called as a primary yolk sac. It is a primary yolk sac. Later on, this primary yolk sac will give rise to secondary yolk sac. Okay, later on, like what happens is that even these cells, okay, these cells, this is called as Husser's membrane. Husser's membrane. So these cells also start producing some connective tissue. Now I hope you understand this, that suppose, try to understand, suppose I say that this is I'm showing it here. Suppose this is the hypoblast layer, okay, cuboidal cells are present here, and this was the layer which was lining the blastocele. Now I am saying that this layer starts producing some connective tissue surrounding it. So if it starts producing connective tissue like this, do you realize that it is going to push the trophoblast far apart? Yes or no?
I don't know.
Okay. If the Husser's membrane is producing connective tissue surrounding it like this, who is producing it? These are producing it, na? Are you understanding this?
Yes, sir.
So if
Yes,
this
sir.
is producing, if you look at the previous picture, is it understandable that it will push trophoblast also farther apart?
Yes, sir.
Right. Now, even how much will it push? Even that also has a limit. So when that limit is crossed, what happens, you know? These connective tissue starts to spread. See, suppose these are the columnar cells. Okay, suppose these are the columnar cells, and this is the amniotic cavity. Now what happens is these cells, they spread on this side. Okay, these cells, they spread on this side. Understood? They
Yes,
start
sir.
to spread on this side. Now if they spread on this side, do you realize that trophoblast is pushed all around like this apart?
Yes, sir.
Yes. So they are pushed like this. Now if they are pushed like this, so now after this what happens, I'll tell you. After this a very strange thing happens. What is that strange thing? Now after this there are some cavities which develop. Like suppose this is the trophoblastic layer, and let's say this is the epiblast, amniotic cavity, hypoblast, primary yolk sac. Now what happens here? There will be some tiny cavities developing like this. So cavities are developing where? In between the connective
Connective
tissue.
tissue.
Acha, in general, what is the common sense telling? Connective tissues are derived from which germ layer? Out of the three germ layers,
Mm.
ectoderm, mesoderm and endoderm, what do you think is connective tissue derived from? Which germ layer? Can you tell?
Ectoderm থেকে হবে নি?
Nah, it is mesoderm mostly. So remember this, ki if we talk about the connective tissue, you can, this is an easy way to remember actually. If you can correlate connective tissue with mesoderm, so remember that this is also called as mesoderm. But is this mesoderm a part of embryo? No, because embryo is that sandwich, red and the green. Embryo means epiblast and hypoblast, that is embryo. So is this mesoderm within embryo? Is this mesoderm present inside embryo or outside embryo? Can you tell? This mesoderm is present. What is embryo? Embryo is basically this area. ठीक है, माने this
Yes,
like
sir.
entire area, this thing is going to be embryo, not right now, but going to be embryo. So this mesoderm, is it a part of the embryo or it is outside embryo? This mesoderm, what I have labeled. What do you think?
Sir, একবার বলুন।
Same question. I am saying that this mesoderm is present inside embryo or outside embryo? Same question.
Outside embryo.
Outside embryo, correct. That is why we call it as extra embryonic mesoderm. ठीक है?
Yes.
Understood?
Yes, sir.
এইটুকু বোঝা গেছে তো? All right.
Yes, sir.
So if you have understood this much, now we can take the story ahead. So you have seen that there are so many tiny spaces which are developing. So these tiny spaces which are developing over here, so what happens to those space? Let's see. Now what happens here is that, see, this is the trophoblast, and okay, this is the trophoblast, and the spaces which were present, okay, this is your amniotic cavity and the epiblast, and this is your hypoblast and the primary yolk sac. Now what happens is that the spaces which were present, they actually coalesce. The spaces which were present, they actually coalesce to form one common space over here. One common space. And this space,
Yes, sir.
you see, one position is there where inner extraembryonic mesoderm, see, if we talk about this space, can I say that this space is dividing the extra embryonic mesoderm into a inner layer and outer layer? Yes or no?
Yes, sir.
Yes. So it is dividing it into a inner layer and a outer layer. So inner layer can be called as visceral layer, outer layer can be called as the parietal layer, but at one place it is connected. See, in this area, can you understand? This is the area, or I can show it with a different color maybe, that this is the area. See, I am changing color, but the material is same actually. This is also the connective tissue, extraembryonic mesoderm, but here, is it separated between the visceral layer and the parietal layer? What do you?
Yes, sir.
Yes, in the blue color which I am shading, is it separated? আলাদা হয়ে গেছে কি? Can you distinguish here between outer layer and the inner layer?
না স্যার, এখনো তো ডিস্টিংক্ট হয়নি।
হয়নি, because ওখানে cavity নেই, there is no cavity over there, right?
Yes, sir.
So this part where there is no differentiation between the outer and the inner layer, this part is called as connecting stalk. Remember this. Understood or not? This is called as connecting stalk. Later on, this is something which will develop into your umbilical cord. Right now it is not
Yes,
umbilical
sir.
cord. Right now it is connecting stalk, but later on this will develop into the umbilical cord. Are you understanding?
Yes, sir.
So this outer layer which is present, this one which was parietal layer, it is called as somatopleuric. See, outer layer, the parietal layer is somatic layer, and visceral layer is planktonic layer. So somatic layer is called as somatopleuric extra embryonic mesoderm. And the visceral layer which is present, see this is all visceral layer. So this visceral layer which is present, this is called as splanchnopleuric, splanchnic layer. So splanchnopleuric extraembryonic mesoderm. And okay, so understood or not? Tell me this much.
Yes, sir.
Right.
Yes, sir.
So this is the status which is present right now. Now, if we forget the outer, acha, at this point when I'm telling the story, এটাও বলে দিচ্ছি যে see what happens now. A part of the yolk sac, okay, a part of the yolk sac is again going to pinch off. Don't worry, I'll show it to you. So like suppose this is the thing which is present, and if you understand, this is your hypoblast, this is your epiblast, this is amniotic cavity. Now observe the yolk sac, what it is doing. Yolk sac is going to pinch off like this. See if you can understand or not. Can you see?
Yes, sir.
So yolk sac is pinching off, and acha, this common cavity is called as extraembryonic coelom. This is called as extraembryonic coelom. Coelom is a body cavity lined by mesoderm. So here it is lined by mesoderm, but it is extraembryonic mesoderm. So coelom is also extraembryonic coelom. ठीक है? Makes sense? You remember coelom, right? Body cavity lined by mesoderm.
Yes, sir.
हाँ.
Yes, sir.
So if I am saying that it is a body cavity lined by mesoderm, so this is a cavity which is lined by mesoderm. But this mesoderm is extraembryonic mesoderm, not the real mesoderm. So the coelom is also not real coelom. We call it extraembryonic coelom.
Yes, sir.
Terminology is logical, na? You don't have to mug up. That's what I'm trying to make here. See, what I'm right now making is the connecting stalk. Since you've understood, I'm not changing the color, but yes, this common area is the connecting stalk where it is undivided. Undivided extraembryonic mesoderm is the connecting stalk. Later on, will form umbilical cord. Okay. Now.
Yes, sir.
Yes. So now, moving on. So I hope now you understand that what happens next. If you guess, then also you will understand. This is very simple. So what you will see is, imagine this is your epiblast, this is your extraembryonic coelom, and this is your hypoblast. Now what happens is that the tiny amount of primary yolk sac will just pinch off. So what will happen? It is going to go like this. It will just pinch off. বুঝতে পারছি কি পারছি না?
Yes, sir.
Okay. So this is going to pinch off. So the old yolk sac, can we call it any more primary yolk sac?
No, sir.
No. That is why it is called a secondary yolk sac. So simple. So what will happen to that thing which pinched off? Nothing. It will just mix up with the coelom in future. It will come down, down, down. It will basically sink. ডুবে যাবে. It will simply sink and it will just join with the coelom. It will just release its content inside the coelom. বোঝা গেল? It will just go down.
Yes, sir.
It will just go down and it is going to mix it here. Okay? So this will be called as the secondary yolk sac. So this is how primary yolk sac got converted into secondary yolk sac. Very simple. तो चलो ये वाला this कहानी এটা হয়ে গেল. Next we will talk about embryo. That is our prime discussion actually. So if you look at this entire structure, whatever I don't know 1 2 3 4 5 6 7 8 diagrams I have drawn over here, but all are 2D picture. What I want you to understand is that this game is not a two-dimensional game. This is actually a three-dimensional game. How? See, when I say that this columnar cells are basically the epiblast, and when I say that these cells are basically the hypoblast, what you should visualize always is that actually there is present a disc, there is present a circular disc which is epiblast, and there is also present another circular disc like this, which is a hypoblast. Basically this is basically a cross sectional view, a cut view. Like suppose this is the plane in which you have cut it, it will come up like this. Do you understand?
Yes, sir.
Yes. So this is basically a disc, it's not a line, it's 3D. Achha, if this is a disc, then what is this cavity? This will be surrounded by a cavity, na?
Yes, sir.
So what is this cavity with the red
The
dotted
amniotic cavity.
amniotic cavity? Very good.
Amniotic cavity.
And what is this green cavity now?
Yolk sac.
Which yolk sac?
Secondary yolk sac.
Yes, now it is secondary yolk sac. Correct. So, and what is your common sense wali understanding? If I ask you that this is surrounded by which layer? This is surrounded by which layer?
Which layer means? The connective tissue there.
What is its name? Do we call it connective tissue? What do we call it?
What? আচ্ছা আচ্ছা স্যার ওটা স্যার সোমাটোপ্লুডিক এক্সট্রাএমব্রিয়নিক ওটা তো
ইমিডিয়েটলি পরেরটাই সোম্যাটো। ইমিডিয়েট সারাউন্ডিংটা কি?
আচ্ছা 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 ইমিডিয়েট স্প্ল্যাঙ্কোপ্লুডিক।
স্প্ল্যাঙ্কটোপ্লুডিক, yes. Then there will be another space that is called as extraembryonic coelom. Yes. And তার বাইরে what will we get?
তার বাইরে সোমাটোপ্লুডিক।
Yes, তার বাইরে সোমাটোপ্লুডিক, yes. And there is only one place যেখানে somato and splanchno they merge together. It is called as somato and splanchno. Where do they merge?
স্যার ওই স্টকের কাছে।
স্টকের কাছে না, that is the stock. ওটাই stock, that is called as connecting stock.
Okay.
ঠিক আছে? And if somebody asks you what is surrounding this?
Trophoblast.
Yes. So you should remember that even this is surrounded by basically trophoblast. And if you have studied, you will know it that first there comes up cytotrophoblast and then becomes syncytium. So that is syncytiotrophoblast. Have you studied that? Cytotrophoblast? And then
no, sir.
outside? No, not yet studied. You will study it. If you don't understand, let me know. So that is called as syncytiotrophoblast, like this. ठीक है? बुझा गया?
Yes, sir.
Right. And outside that, what is present? Outside that, endometrium is present, which is called as by now decidua. You'll learn all these things. I'm in the 3rd week of development, so that happens in the 1st, 2nd week during implantation time. Okay, so let's come back to our discussion. So what happens is, let's focus on the disc now. So if I talk about this disc over here, the epiblast, okay, let's focus on epiblast for a little bit of time. So this is epiblast. So if this is epiblast, see in this all these columnar cells, they are held to each other. Do you agree or not? They are, somebody is holding them together,
Yes, sir.
right? There was gap junction, but gap junction cannot hold cells together. Somebody must be holding the cells together. So question is, who is holding the cells together? So remember, in between them there is a glue present. In between every cell there is a gluey substance. Okay, what is this gluey substance which is holding it together? This gluey substance is called as E-cadherin. E-cadherin. Okay, E-cadherin is a protein which is actually holding these epithelial kind of cells. See, adhere, and here it is cadhere. You can correlate adhesive, adhere, chipku,
Yes, sir.
and cadhere. And any protein ends with, any protein will end with
In Y
in, in, in.
in sir in.
So cadhere or adhering protein, adherin. So it is cadherin, not
Mm.
adherin, we call it cadherin. So, and it is working here on what kind of
Yes,
cell?
sir.
Epithelial cells. So E-cadherin. Can you remember this one?
Yes, sir.
Right. So it is E-cadherin cells which are holding these epiblastic cells together. Now, what happens is that here some cells, they start to proliferate. Okay, certain cells are present over here, like certain group of cells are present over here, and you know these cells start to proliferate like this. So when they start to proliferate, they form a streak like structure, straight structure, like
Yes, sir.
lightning. Light, we say na, streaks of lightning.
Mm.
uh, if you see, you know, a lot of people, mostly the females and some males, you know, they will make streaks in the hair. What do we call it? Streak, straight line. Have you heard of this word?
Mm.
Yes.
Yes,
So,
sir.
so we call this one also as a streak, a line. And since it is happening at a very early stage of life, so we can call it as primitive streak.
Primitive streak.
So this is primitive streak. And this area, it forms a little bit knob like structure, and it's a group of cell, like we call group of cell which are which do electrical activity in your heart. What do we call it? Nodal cells. SA node, AV node, we call it node. So even
Yes,
this
sir.
is also considered as a node, so we call it as primitive node.
Primitive node.
Yes, so this is a primitive node. Now, after a while, what do we see? There is another thing, another molecule which starts to express at this point of time. Now, this molecule, which actually has a job of working against E-cadherin, that is called as fibroblast growth factor 8. There are many fibroblast growth factors. So FGF1, FGF2, FGF3, FGF4, this way. So this one is FGF8, fibroblast growth factor 8. Fibroblast growth factor, whenever your DNA will express fibroblast growth factor, okay, in this stage, during the 3rd week, whenever your DNA will express fibroblast growth factor, your E-cadherin is going to down regulate. Understood or not? Your E-cadherin
Yes,
will
sir.
go down. Now, what was the job of E-cadherin? To hold the cells together. Now, if fibroblast growth factor 8 comes up, can the cells hold with each other?
No, sir.
No. So what happens? All these cells, all these cells which are present over here, they are going to simply collapse. because nobody is holding them. So these cells, they are going to collapse like this. So these are the intact cells where still E cadherin is there. Here there was no FGF8. Here E cadherin is still there. But this area, in this area, E cadherin is not there because FGF8 is there. So
Yes, sir.
this is the part where FGF8 starts getting expressed and E cadherin is gone, and hence this cell collapses. Clear or not?
Yes, sir.
Okay. Now, what kind of cells are epiblast? By shape, if you look at the morphology, if you look at the shape, so what is the shape of the epiblast cells?
It's a columnar.
Right, very good. So they are columnar cells. So what happens is, now the columnar cells which are present on the sides, they start to migrate. Okay, so let me show it here in this way, that the columnar cells which were present over here, they start to migrate. Now when they migrate, of course from both the sides they will migrate. Now as they migrate, the cells change their shape, their morphology, and these cells which were as of now, they were what? They were columnar. Now,
Columnar.
now they become flask shaped. Flask shaped. We call it as, you remember this kind of shape? Animal kingdom. Lots and lots of lower animals. So,
Flask shape.
say hydra. Flask shaped.
Acha, acha, acha. Yes, sir. Hydra.
Yes. So, these kind of cells, they are flask shaped. They become flask shaped cells, and they will just dive down. They will just fall down like this. Okay? They will fall down. Now, when they fall down, these flask shaped cells, what happens in the beginning? In the beginning, if you remember, there were these hypoblast cells present. You cannot forget hypoblast, na? Hypoblast is present, na, here.
Yes, sir.
The first step is, these cells, they are going to attack these hypoblast cells. These cells, they will attack these hypoblast cells, and it is going to take its position.
Yes,
And
sir.
it will take its position. It will replace the entire hypoblast and hijack that location. So what will happen? Like, suppose these are the columnar cells. Suppose these are the columnar cells. Now you understand that all the epiblastic cells migrated and they dived down and they displaced the hypoblast and they developed a new layer. Can you understand this?
Yes, sir.
Yes.
Yes, sir.
So can we call it hypoblast anymore?
No.
No. So this is endoderm. So this is endoderm.
তাহলে স্যার এটা স্যার মানে কিউবটাল হবে তাহলে
Actually, shape also changes here. The shape becomes polygonal.
আচ্ছা
Okay.
আচ্ছা
So this is going to be endodermal cells. And even these further more cells, like cells were constantly migrating, epiblast cells were constantly migrating. Now as they are migrating, now what will happen to the rest of the cells? Where will they go? So they are forming sandwich in between these. They are forming sandwich in between the epiblast and endoderm. So this sandwich,
mesoderm তাহলে এটা।
correct. So this is mesoderm. And remaining whatever epiblast cell is left, that is called as ectoderm. This is ectoderm. Understood or not?
Yes sir.
Understood. Great. Now before we proceed ahead, let me tell you কি এই ঘটনাটা কোথায় ঘটছে, where is this story happening actually. So remember, uh, when it comes up to your embryo na, so always you should note that there are few things which are genetically decided. What is it? Genetically your craniocaudal axis is decided. Craniocaudal mane which side is going to be your head side and which side is going to be your tail side. So craniocaudal part is decided. Even your left and right is defined. I understand. It cannot happen that it is random.
Okay.
Like somewhere it is head is forming, somewhere tail is forming. It is not like that. They are very much they are specified. And remember, if somebody asks you, head and tail is decided first or left and right is decided first, the answer is head and neck. So first comes up craniocaudal axis, then comes up left and right axis. Okay, so actually what happens is
Yes, sir.
that this endoderm which is forming na, so basically try to understand one thing. This endoderm, as you can see in the diagram, it is going down like this. It is going down like this. What you don't
Yes, sir.
understand with the diagram is that they are not going down, they are also migrating in. See, when something is going down, it is called as invagination. My point is, they are not only invaginating, but they are also spreading. Can you understand this? They are also spreading
sir.
like this.
Yes sir.
Yes, and if you understand that they are spreading, do you also understand that they are not only spreading two-dimensionally, they are spreading three-dimensionally as well?
Yes sir.
Like this, like this. They are spreading three-dimensionally, like this.
Yes sir.
And they are moving more cranially. So this kind of endoderm, if you talk about the endoderm, like if I say the endoderm which is present on this side, the endoderm, mind it, it is called as anterior visceral endoderm. Okay, in the bilaminar disc itself, it is already decided that from the bilaminar disc, when endoderm will develop, because they are gradually moving towards trilaminar disc. So in that bilaminar disc, the endoderm which is present, they become very specific. The endoderm towards the head area, that is very specific. It is called as anterior visceral endoderm. Okay, so why?
Yes sir.
How come it is? How is it developing? Because this, why do we call it as specifically? We have given it a special name. Why? Because this actually expresses some special gene. So there are some special gene like say OTX2, okay, LIM1,
Yes sir.
and HESX1. Okay, so these are certain genes which are expressed from this. What are these? These are some genes. These are some transcription factors, to be very precise. These are some transcription factors. So these transcription factors, they are released from this anterior visceral endoderm. That is why this part of endoderm has been given a special name, because they do a special job over here. Okay? Understood?
Yes,
and there
yes,
are
sir.
some secretory factor also. See, head. What is present towards the head? Cerebrum. So also you can remember this, that there are some secretory factors also. So what are the secreted factors? See, this is cerebrum. Secreted factor is cerebrus, or actually the spelling should be cerberus, but it's easy to remember, cerebrum cerberus. And also there is another one that is called as lefty one. Okay, so this one, this lefty one, these are all secretory factors, and they are released from always the cranial end. মানে যেখান থেকে Cerberus, lefty one, OTX2, limb 1, hesex 1 যেখান থেকে প্রডিউস হচ্ছে, that is going to be the head region.
Cranial end.
That is the cranial end.
Yes,
Correct.
sir.
Very good. Okay. And absence of Cerberus, absence of lefty one is going to be the caudal end.
Caudal end.
বোঝা গেল? Now
Yes,
wherever
sir.
Cerberus lefty one is not present, সেইখানে primitive streak will form. অর্থাৎ if somebody asks you that primitive streak is forming towards the head end or the tail end, head end or the cranial
Tail
end
end.
or the caudal end, so what will you say?
Caudal end.
Yes, very good.
Caudal
So
end.
suppose I say, very good, so if I say this is cranial end and if this is caudal end, so cranial end cannot have this primitive streak because there is lefty one Cerberus present. So on the caudal end where this is not present, there we are supposed to see this kind of groove. So it is always present on the caudal end. Okay?
Yes, sir.
Right. Now once the streak is formed, here there will be present some special group of cells which are called as nodal cells. Nodal cells. ठीक है ना? And these nodal cells are going to be responsible for creating dorsal mesoderm and ventral mesoderm, and there are different other types of mesoderm. So all these things they are going to produce. Okay, so this, I hope you have understood that this nodal tissue, this is very important, that this node is actually doing a very important task. So if node is present, primitive streak will be present. Simple. If nodal tissue is present, primitive streak will be present. But if lefty one and Cerberus is present, will nodal tissue be present?
No, sir.
No. So like, there are a lot of things which are present. Like suppose I try to show you the side view of it. See, now you are learning at a medical level, so these are certain things. I think this is interesting also. These are certain things which you should try to know. So you see that suppose I say that this is your so-called side view of your ectoderm or epiblast. Okay, basically I am trying to take a section sort of like this. Right? I'm trying to take a section like this. Can you understand?
Yes, sir.
This kind of section, side view. We are looking from the side view. We are looking at it. So if this is your ectoderm, that means niche somewhere there will be endoderm also. Now what we learned just now is this area, suppose endoderm, this is expressing hesex1, OTX, the limb, those genes. Okay, it is Cerberus, lefty one, all those things it is expressing. So what do we call it? This shaded area, this part, this is what? This is anterior. What is this becoming? This is becoming anterior visceral. Yes,
Visceral ectoderm.
Anterior visceral endoderm. Yes. So this area is becoming anterior visceral endoderm. Now remember, there is another thing present, another factor present. It is called as bone morphogenetic protein. Now remember that bone morphogenetic protein is secreted throughout the bilaminar disc, and it combines with FGF. You remember FGF8? FGF8 was actually creating that gaddha, converting primitive streak into the primitive groove. So it combines with FGF8 to ventralize the mesoderm. Okay, so how will the mesoderm go in? So how will it go more in? So that is because of this BMP4. So even remember that there is thing called as BMP4 which is secreted throughout, and if somebody asks you that what is its benefit. So later on you will read, it's very interesting. Right now maybe you won't be able to correlate, but for the timing just trust me that there is a, from this mesoderm also, from this part there are certain fragments of mesoderm which develop. Now out of that one fragment gives rise to your kidney. Okay, mesoderm will further divide later on. Okay, later on you will read that mesoderm is further dividing. So there is lateral plate, there is intermediate. Okay, so we have names for that. So we will discuss that later on, not right now. But what happens is each part, like if I say intermediate mesoderm, if I say lateral plate mesoderm, if I say paraxial mesoderm, every mesoderm has their own work to do. Like I said, that one such mesoderm is responsible for your urinary system, the kidneys. So you see, unless and until this mesoderm is going ventrally, unless and until the mesoderm is ventralized, do you think your kidneys will form? Kidney is actually found from a thing called as
No, sir.
intermediate mesoderm. So if your mesoderm never goes never goes deep down and develop intermediate mesoderm, do you think your kidney will form?
No, sir.
No, yes. So this is true. So it is the job of BMP4 to push down every single thing, every single what do you say, every single like mesoderm downward. And reality is that suppose somewhere I do not want mesoderm to go down, so I need to inhibit BMP4, because BMP4, what did I say, BMP4 is expressed throughout the bilaminar disc, everywhere BMP4 is present. तो बेसिकली उसका बस चले वो जो चाहे एंटायर जिनिसटाके नीचे नामिয়ে দেবে. So wherever you don't want कि जिनिसটা नीचे नामुक, so in those cases you have to inhibit it. সেই জায়গাগুলোতে where you don't want mesoderm to come down, you actually have to inhibit it. আর সেই inhibit কে করবে? Once again this nodal organizer. এই জন্য এটাকে organizer, some people also call it as organizer, nodal organizer, because its job is to organize. Okay, this node itself is the organizer. So
Yes, sir.
so there is this, it's a famous person, Hans Spemann. So it's a Speman's node or Speman's organizer. People call it as Speman's organizer or Hans organizer. Okay, so he actually described this. Okay, so now if I want to say, if I want to talk about that the nodal area or everywhere where there is BMP4 present, so that means everywhere it is just coming down. So that means if you have to create dorsal mesoderm, all of these are ventral mesoderm. If you have to create dorsal mesoderm, how to do that? BMP to সব জায়গায় করে দেবে. So you need to inhibit it.
Yes, sir.
So BMP, if there is BMP4, so BMP will ventralize all of everything. It will try to ventralize everything. So somebody has to be there to stop this person. So
Yes,
who is this somebody? Who will stop this person? So here there are some special proteins which are present. Okay, special factors which are present. So who is doing this? Node. Node has to stop it. So what is node doing? Node is actually releasing glucocorticoid. It is releasing chordin. It is releasing noggin, follistatin. Okay, so all of these things, they are going to be they will antagonize BMP4 activity. So you understand how dorsal, ventral, cranial, caudal, they are all predetermined. Are you understanding?
Yes, sir.
Yes.
Yes, sir.
So actually, there are lots and lots of genetics present over here. There is a brachyury gene also present at the node. Okay, so I'm not going with those things about those brachyury, T-box. There are lots and lots of genetics present. We are just discussing basic stuff. Like you have to know, you should know as a doctor that how your cranial end and caudal end is differentiated. Which becomes your pete, which becomes your pate. What is your dorsal ventral? Lateral, done, bum. How is the right and the left coming up? Okay, so what happens is that sometimes, let's say if goosecoid is present, goosecoid is not going to allow this differentiation between dorsal and ventral. So what happened is, there are a group of doctors, what they tried is, in a frog embryo they actually overexpressed goosecoid. Okay, I don't know, maybe it will sound interesting to you. What some doctors did, they did something so that goosecoid gets overexpressed in the frog embryo. You know what was the result? Differentiation between dorsal ventral or that thing never happened. And since it never
Yes, sir.
happened, so because of that, there was two-headed tadpole which was born. Understand? Two-headed tadpole. So, this is the importance of all these things. Understood or not?
Yes, sir.
Right.
Yes, sir.
So now let us proceed ahead. So, discussion is becoming, I don't know, I am finding interesting. Are you finding interesting? No. Confusing.
Yes, sir.
Acha, okay. So now, okay, we will take a pause over here and we will learn now something new, that there is something else happening here. What else is happening? So this time, let us take a section. So, acha, okay, let us take a section here. Here, we'll take a section like this. Okay, understood? We'll take a section like this. So if we take a section like this, how will it look like? It will look, I don't know how will it, is it looking something like this? Is it understandable?
Yes, sir. Yes, sir.
See, I want also you to know this, that as cells migration is taking place on the caudal end. Try to understand this. Cell migration is taking place only at the primitive streak area or the primitive groove area, and that is present where? That is present in the caudal end. So all the cells from the sides in the caudal end is going towards the medial side. Yes or no?
Yes, sir.
And when
Yes,
they're
sir.
going medially and they're dropping, they're moving anteriorly, they're moving head side, cranial side. Yes or
Hmm.
no?
Yes, sir.
So can I say that the head side is expanding and the caudal side is shrinking? Yes or no?
Yes, sir.
So what I'm trying to say is your disc, will it remain circular or it will become pear shaped?
Pure shape, sir.
So remember, by now when we are discussing this, that is why I have drawn it in this way. Your disc is going to go pear shaped, because from here all the cells are gone and all of these cells, they are all spreading in this direction. So now in this picture also, you see I am showing it all on the front and the lateral, anterolateral, but not anterior. Do you notice that? I am showing all of these things on the anterolateral side. So all the cells are migrating anteriorly, but laterally also, not straight anterior. Can you see that there is, are you understanding, there is no cell which are migrating as of now, whatever I taught you, there are no cell which are going straight like this. Yes or no?
Yes, sir.
Yes. So now I'm going to explain it, that even some cells are going to drop like this and they will spread anteriorly, but they are going to be different. That is why I'm going to discuss it separately. So these cells, as they are going to go down, okay, so initially what will happen, initially they are going to form a structure, they will form a tunnel like tube like structure. So here there will be gradually a tube formation. Okay, like suppose if I take the same picture and I try and I want you to see it uh, from the cross sectional view, not the longitudinal section. I want you to see it from a cross sectional view. So how will it look like? It is going to, so you see, if this is the case, so what I'm trying to show you here is that the cells, the epiblast cells, they will form a tunnel over here like this. Try to correlate. Tell me whether you understand or not. Correlate both the views. Endoderm, endoderm. Yes or no?
Yes, sir.
Yes. Okay. So, of course, and if you correlate, what I'm trying to say is that right now we are seeing the cells going vertically, like, not vertically, like anteriorly straight, but on the sides there are still mesoderm. Anterolateral movement already has taken place, isn't it? So I must fill this
Yes,
part
sir.
with mesoderm. Yes or no?
Yes, sir.
Yes. Okay. Thank God you are getting it. Okay. Now what happens is this cell is, this is a cord. This is a tube. Now what happens? This tube opens up. Now, how? What do I mean when I say it opens up? So basically, suppose this is the idea, and this is the thing. We have taken the section. We have taken the section at the level of primitive pit level. Now, what I'm trying to show you here is, acha, let me draw this one as well, and even this one as well. So now what happened is your your cell is going to open up like this, and next step is it is going to collapse like this. So your epiblast is going to remain like this, your hypoblast will remain as it is, and your mesoderm is going to go like this. And yes, now congratulations, you have got the notochord. This is called as notochord, our discussion of today. So finally we created the notochord. Have you understood or
Yes,
not?
sir.
Okay. So because, so that means if I am going to talk about the entire, let's say the disk, like, we started over here. So what I'm trying to say is that the notochord, that means in this picture, this notochord will be going vertically like this here. Understanding? Where I have put question mark earlier, actually that is not the question mark here, that is the area where notochord will be present. Understood or not?
Yes, sir.
Right. So this is notogenesis. Okay. So uh, today we have done a lot. I think you've understood this part, whatever we discussed,
Yes, sir.
because we want to do neurulation today, how neural cord forms. So that we will discuss in the next class.